I'm very happy to introduce Dr. Phil Townsend for our seminar today. Uh, Phil got his bachelor's at University of Virginia, and I believe it's sometime after that he said he was drawn to science or interested in science because of a failed music and writing career. He then went on to get his PhD at the University of North Carolina. After a couple stops at Texas A&M, Maryland Appalachian Labs, he ended up at Wisconsin-Madison, where he's been ever since. Phil, I always like watching Phil give talks. He's emerged. He is a leader in remote sensing of plant physiological ecology and tells really amazing stories, known globally, over 100 papers, 15,000 citations, all the metrics, to hit all of that stuff. One of the things, he's a very nice person. One of the things he's really interested in is how his lab and his program have developed open access data in open access data tool platforms. And an example of that is ECOSYS, or the Ecological Spectral Information System, which is an online repository for spectral data and other paired metadata that can be put together. There's a number of other platforms and tools that you can find if you go to his website on GitHub. So if you go there, that's one thing he's exceptionally proud of. Outside of all of really the amazing science stuff that Phil does, he's a really good person. And I say that I've known Phil for a while. He was on my PhD committee. He actually funded the last year of my PhD to kind of help out with projects that he had going on in his lab. And then I ended up staying as a postdoc and scientist in his lab, I think for 5 years before I came here to Purdue. And the one thing that I will say is Phil is an excellent mentor, right? Whether he knows this directly or indirectly, watching him or him explicitly saying things, he prepares a lot of his students to go out and really be successful in the job market. And a lot of the individuals that come through his program are highly successful in places like JPL, R1 universities, and contribute a lot to kind of the idea, a lot of the concepts that Phil's going to talk about today. So with that, I'll turn it over to Phil.
Didn't mention my basketball playing ability. Yeah, so I have listed names of people here, and I realize that there are a few people whose names aren't here, but all of these people who are listed here are people who've contributed in a really meaningful way to what I'm going to present here. So I'm going to warn you, and this warning that I'm going to give you right now is actually asking you all to give me a warning. When you're starting to get tired of me, let me know, because I had a presentation that I gave several months ago that was stuff over the past few years, and seen that. And then I started looking through it and I thought, oh, we've got all this new results here. Let me weave this in. And so then I started deleting slides and putting slides back in, and it became this kind of octopus of a presentation. So I don't have, it's a series of vignettes. I don't have a good sense of how long it is. I recognize that I might end up going a little bit long. Just stop me. You can't hurt my feelings. I was working on it this morning to try and make sure that I had it within time, but there's a lot of content that's in here. So I apologize in advance for that. Okay, let's see if I can advance my slide. Okay, so what do we do in the Townsend lab? And here's, I'm illustrating my presentation with a series of paintings from David Hockney, a British-American painter who has wonderful outdoor landscape paintings. So what most people think of me as doing is airborne imaging spectroscopy or hyperspectral remote sensing. We fly sensors on an airplane and also on a heavy lift drone to collect imagery over a lot of different settings to understand patterns of plant traits. But it's not just the remote sensing. is actually underneath everything that I do. And the reason why I have all these different collaborators and work with different people in different areas is that we study ecosystems in a lot of different ways. We look at leaf level processes, we sample leaves, we sample plots in the field. We work in experimental settings with experimental designs where we can make measurements. We make measurements in the field, we make measurements in the lab, we do analytical chemistry, we fly airplanes and drones. We work with data from, say, NEON or other people who fly airplanes, with the ultimate goal of being able to scale up the insights that we're getting globally once there are global hyperspectral satellites. SBG, which is the NASA one, 2032, that's really awfully close to the end of my career, but it's been something that I've been working on. And when we have it, I hope that I'll be able to convince you that we'll be able to transform our understanding of the world. And so ultimately, for me, it boils down to maps of foliar traits. And my next slide, I think, says it's actually more than mapping traits. But this is just an example of the sorts of things you'll say. This is from NEON. You're seeing 3 different traits over a whole bunch of different NEON National Ecological Observatory domains. And I'm showing 3 foliar traits, chlorophyll, phenolics, and LMA, in these ternary diagrams in these different colors. And so across all of these different biomes of the US, you can see a lot of variation in the traits that are driven by environment, the types of species that are there, and so forth. And you can also see variation in the diversity of those traits across these sites. So this is kind of going to be the fundamental background. So just a little bit about NEON. It's all these sites. There's 68 or 70 terrestrial sites that get flown with an airborne hyperspectral imager every year. We were funded, John was on the project and was on the main paper that's come out from this to develop trait maps from all of this. So NEON collected the imaging spectroscopy data, we made the trait maps, and as part of that, we actually sampled across all of the domains except for one. The one domain we didn't sample was Hawaii, which we're going to actually sample this year, even though the project is over. We're going to get there. And then we develop models. I'm actually not going to talk about the methods at all. I'm actually going to talk about how we can use the outputs. There is a lot behind the models. So this is actually just showing our predictions on a nationwide basis of nitrogen. This is with the independent data, the data that's withheld from the model, and how the model performs across different plant functional types. But I'm not going to actually show a lot of figures like that. I'm actually going to concentrate mostly on how we can take this trait data and learn more about the ecological functioning of our ecosystems. So back to trait maps. So different traits across different sites gives us insights into what's going on that might be different between, say, Guanica, which is in Puerto Rico, and Abbey Road, which is in Oregon. And one of the interesting things, and this is just showing 3 of the 24 to 30 different traits that we show, is that different traits have different importance in different areas, in different ecoclimatic domains. Okay, so like I said, we're making all of these maps. What my talk is going to be is about more than just mapping. It's actually going to be about the use of these trait maps to understand plant biology and ecosystem function. But I am going to give you a little bit of a background on imaging spectroscopy and traits. So we've known for a long time that the reflectance off of leaves is related to the chemical composition of leaves, and this changes based on the composition of the species, the phenological state of development, and so forth. So you'll see a lot of figures that show wavelength on the x-axis and then reflectance on the y-axis. And this is just showing papers from the 1920s, 1929, where some of the first publications that came out about this. And this is just seeing chlorophyll absorption features and green hump, plants are green, and so forth. But there are lots of features in plants that are associated with traits. And so if you look at this, this is a leaf level measurement. Most of what I'm going to be showing you is going to be from remote sensing data. You can actually see that there are all sorts of features especially in the wavelengths outside of the visual wavelengths that are related to the chemical composition of leaves. Our whole process of mapping traits is identifying those features. And so what hyperspectral remote sensing does in comparison to, say, multispectral data, where you might just sample several different wavelength regions, is you actually measure the whole spectrum. Why do you measure the whole spectrum? It's because there are features like this one right here that you use to map particular traits. Maple, phenolics. Maples have high levels of phenolics. I'll talk a little bit more about that later. So to use an analogy, we're looking at the large crayon set, and each one of these different colors of the crayons, or the variation that you might have in it, is related to traits that you wouldn't necessarily be able to characterize with the standard remote sensing data sets. And the way that you might visualize it, this is all the same image. This is from NASA's AVERIS NG airplane sensor. We can show it in the visible wavelengths. We can show it in the color infrared composite, or we can do some transformations of the data. In this case, it's what's called minimum noise fraction, and then find combinations that actually can bring out a lot of the variability that we might be interested in, all from the same data set, trying to illustrate the range of variation that you might see. So when we work with an image, every pixel, this is an image cube. Those of you who work with imaging spectroscopy see these a lot, but think of it as every pixel right here is actually a spectrum that you would measure and that we use, and we're interested in finding those little features that are there that are related to plant traits. So plant traits. So that's as far as I'll go on the imaging spectroscopy. A whole lot of math and a lot of methods behind how we analyze the data and process the data that I'm not going to go into. I'm just going to talk really briefly about foliar functional traits. So first, we'll just talk about what the function is. Photosynthesis, building of structure, the vertical structure of plants. Plants will do respiration, they'll grow, they'll reproduce, and they'll defend themselves. There are other functions that might be present there as well, but this is basically the functions that we're talking about. And then from remote sensing, we can measure some traits. There are other traits we can't measure. And this isn't showing all of the traits, but this is showing some of the traits that we get from imaging spectroscopy that we would be able to use. So chlorophyll and other pigments and nitrogen, very strongly related to photosynthesis. Photosynthesis happens, creates non-structural carbohydrates. Non-structural carbohydrates, sugars, and starches become the building blocks for everything else. Building leaves, lignin, cellulose, phenolics, and other defensive compounds, and a whole bunch of other things that aren't shown here. There are other elements in addition to nitrogen, such as phosphorus, calcium, potassium, that all play important roles that are also somewhat to moderately detectable using remote sensing, and that are related to nutrient availability and so forth. So this is sort of what we're basically trying to characterize. These are the traits. These are the functions. So the way I'm going to illustrate how we use this is through a series of vignettes related to functional traits across diverse biomes, such as all of the different types of neon, looking at seasonal trajectories, looking at responses to the environment, such as drought or insect defoliation, and then the way that we can use this for monitoring something like GPP, like a function right here. We use functional traits because we can't map all the species. There are many thousands of species that occur in a canopy in North America, for example, and 350 or 400,000 unknown number of species planet-wide. So we concentrate on functional traits and assume that the diversity of functional traits is somehow related to the diversity of ecosystems. So I'm just going to start with just a brief vignette, first of all. So two NASA campaigns. One is in Bioscape, which is in the Mediterranean ecosystems of South Africa. The other is above, which is the Arctic boreal area of North America. And actually, as you look at these pictures of these two ecosystems, you see things that look a lot the same. You don't recognize that this is the most diverse in terms of species richness, most diverse ecosystem on the globe. Whereas this has just a very small number of species, but actually the functional diversity here is quite a bit higher than the functional diversity here. The systems are driven by some similar and some different characteristics. So the same functional traits may not be important for both of them, but these two ecosystems are both very important globally, kind of look the same in many respects, but have very different characteristics. And somehow we need to kind of tease that out. And I'm sort of wanting to make the case for using traits rather than using the spectral data in and of itself. One of the things, so this is just based on spectral data, not based on traits. So hyperspectral data, you have hundreds and hundreds of wavelengths. You can basically do something like a principal components analysis, and you can look at the scree plot of the amount of variance that's explained and find a threshold to identify where you're at a noise level. And what we're seeing here, if we look at above, which is the Arctic, and bioscape, which is South Africa, that actually, when you look at the data over here, the above actually looks like it's more diverse. It has a higher intrinsic dimensionality, which is this concept from Kerry Klaus-Nicholson, than you have over here with bioscape. So one of the things that's telling you is that if you just look at spectral data, you're not actually getting the whole story. And if you translate that into the traits, you might be able to learn a bit more. So that's what we did. So we can go this is actually showing the spectra. Again, this is actually showing the data a little bit different way. This is showing the spectral space, so a principal components analysis done on all the spectral data. And you see above in blue, bioscape in the orange, and also I've thrown in the neon data as well, just for background. And you'll see that there's not a whole lot of difference in terms of the spectral space and the amount of space that's filled. So let's look at the traits, though. So again, I'm using neon just as sort of background information. Neon has lots of different biomes, including Mediterranean biomes that you would see in bioscape, although not neon is just North America, it's not South Africa, and above. And you'll see that there are different ranges of variation. So for example, bioscape is a very South Africa, very low nutrient ecosystem. On the other hand, you look at phenolics, which are defensive compounds, very important in ecosystems that have high insect pressure and that are also seasonally stressed due to water stress. You'll see a lot of that. You'll see a lot of these variations. And so and remember, phenolics, very narrow spectral feature at 1660 nanometers is what we use to detect phenolics. You do a principal components analysis on spectral data, that's going to be buried in your dimensionality analysis. So this is where you actually start to see some differences. And this is just two different sets of traits. This is basically traits that are related to pigments and everything else. You actually start to see that the Arctic boreal and the bioscape data from South Africa start to separate out from each other, start to cover different areas of spectral variation. And you'll see that the Arctic boreal and neon kind of cover the same sort of area. And that one of the reasons why South Africa is so diverse is because it's got a different trait space that's filled when you start looking across all of the different traits. And so this is just showing kind of the same data, but boiled down a little bit more, where you actually start to see that the South African data actually fills a much larger trait space than the Arctic. Even though there's a lot of functional diversity here, there's actually a lot of trait variation. One of the really, really cool things that came out of this is that we can estimate isotopic ratios from the hyperspectral information. And you can ask me questions about how we do that and why we can do that later. But this actually completely makes sense. South African ecosystems are water stressed. Carbon-13 carbon is used largely as an indicator of seasonally integrated water stress. N15 is largely used for nutrient availability. You see this gradient between the Arctic and South Africa of higher nutrient availability, higher water stress over here. So this gives us an insight at a sort of a pseudo global scale that makes sense. Okay, so let's go back to the NEON data. I'm going to show you just a series of slides through all of this where I just use the NEON data as a background. All I want to do here is just point to, we can look at trends of traits. We could look at trends of trade-offs between traits. So if you have high nitrogen, you tend to have lower leaf mass per area, for example, so leaf thickness. And all I'm showing here is the correlations between leaf mass per area, so investment in leaf structure, versus some other traits, non-structural carbohydrates, phenolics, water, and nitrogen. And here now it's separated out by species. The species data came from work by from NEON's own sampling data. And what is important to show is that the relationships differ among different species. So there's no one broad generalization as to what might drive trait-trait relationships. Some are really stable, like the relationship between LMA and phenolics. We'll talk more about whether that's true or not in just a bit. And some of them are much more variable. And for some of them, species have very, very different relationships. This is really hard to do with in situ sampling and laboratory chemistry. Really, this is the sort of thing that we can actually only do if we can collect imagery and estimate these traits over large areas, because we can't get people out there. We can't pay for all the chemistry and so forth. And Adam Clues, former PhD student of mine, used data from this Prisma satellite that the Italians launched to show that if you look at global patterns of phenolics, which are defensive compounds or used as an indicator of defensive compounds, are much higher in the tropics. This is actually what we would expect from ecological theory. It's been really hard for people to go and actually sample this across all of these different places. We see this relationship. Theory would predict it because there's much higher insect and pathogen pressure in tropical areas than there is at higher latitudes. And so trees invest more in chemical defenses. Similarly, in terms of like something we've not been able to do before, talking about South Africa, this is Cape Point, this is Cape of Good Hope right there. We really, before this project in South Africa, we just really didn't know anything. Here I'm showing nitrogen, phenolic, and non-structural carbohydrates. You'll see some areas of species that are legumes that are very high. Their nitrogen fixture is very high in nitrogen. And you'll see that there are some species interspersed here with more defensive compounds and more in non-structural carbohydrates. And just as a reminder, non-structural carbohydrates are the building blocks for everything else. I'm going to interpret non-structural carbohydrates as this in the foliage as this reserve that the tree or the plants are trying to decide what to do with. So if it's non-structural carbohydrates in the leaves, the tree has not made a decision yet whether to turn that into wood, send it below ground, or turn it into defensive compounds or something like that. Held ready. Okay, that's sort of the first vignette, sort of looking at what sort of insights we get from thinking about traits kind of at broad scales. Now let's think about temporal scales. So Adam Clues, as part of his dissertation, we imaged this place called Black Hawk Island, which is an island in the Wisconsin River. It's actually the first site from a 1988 paper by Carol Westman in Science that showed that you could use hyperspectral data to estimate foliar traits. And actually some of the monumental ecosystem research done by John Aber, Jerry Melillo, big names in ecosystem ecology was actually done at this site. Because there's actually 5 different soil orders present here. And a lot of those people were affiliated with the University of Wisconsin at the time. And so it was easy. It's about an hour north of Madison. So we actually wanted to see how traits varied over the course of the season. A lot of assumptions are made about traits mid-season, summer traits. are representative of how the ecosystem functions year long. We actually wanted to look at that, and so we imaged it 11 times over the course of this one summer. Adam collected field samples, and we did field assays on the samples. And that allowed us to do this, to make a map of foliar traits. We also mapped all of the species on the site, and we could look and see how traits evolved over the course of the season. And I'm gonna kind of go back to this figure shortly. But what you can see is that across these different species that are shown here, that they have different trajectories. And actually, everything that's shown here is a broadleaf deciduous species. There's also some conifers in here and some other species. We just cherry picked the species that are essentially the most common species. What you may have noticed when you looked at this diagram here too, is that they diverged and came back together in different ways, these different species, where they fill the trait space. And this allows us to develop some insights. So we can look at some traits like calcium, chlorophyll, and nitrogen, and we kind of see what we expect. Early in the season, these broadleaf deciduous trees have a lot of nitrogen while they're building up everything else, and then it declines, and then everything is retranslocated out. Chlorophyll follows a trajectory. Calcium is not retranslocated, so all that happens is the calcium is just taken up and accumulates, and it's redistributed back to the forest through senescence at the end of the season. However, you see interesting insights when you look at lignin and phenolics. So phenolics are defensive, using it as an index of defensive compounds. Lignin is a structural compound generally associated with water transport, but lignin is probably the most important and the most recalcitrant structural compound in leaves. And you see some very different trends. So here we've got the two maples. You remember I said that maples are very high in phenolics content. They're very high from the beginning of the season. The other species have much lower phenolic content. And so there's a different strategy over the course of the growing season. First of all, maples have very low amounts of lignin. And if you've been out into a forest, you know those maple leaves, they decompose. The maple leaves fall, you come the next spring, they're gone. Oak leaves, not so much. And so that's the lignin at play. So these non-maple species invest a lot of effort in building lignin, and they have higher phenolics at the beginning of the season. These drop once they have enough lignin for lignin to act as a structural defensive compound. Over this too, but we can actually start to link this now to the drivers of trait variation. So look, for example, species phenolics, the biggest partial dependence is with species, and that really relates to some species being very high, having high chemical defenses. And we can also look at stoichiometry and how that changes over the season. Stoichiometry can give us some information about ecosystem functions related to carbon uptake. And a lot of this has to do with dilution effects. As plants build up more material, they may not actually change the total amount of something that they have, but they'll change the total relative amounts. We did a similar study in 2022 in Southern California called SHIFT. So this is in the Santa Barbara region right here. Later, I'm going to show you some analyses from the Yosemite National Park, southern part of the Sierras right here. This is an area that was imaged nearly weekly over the course of the spring, so February to June in California in 2022. And again, once again, we can look at some very basic variables and how they change over the course of the growing season to maybe provide some insights. So here you see differences between the grasslines and the trees, but really it maybe is a little bit more useful to sort of look at this, look at several time slices from February to May and visualize the patterns rather than just see a moving dot. And you'll see things that you might expect, but you'll see patterns of increasing defense, investment in defense and LMA. So building up structure and leaves, building up more defensive compounds. Here we're seeing nitrogen, LMA, and phenolics. and decreasing nitrogen. And this is there's a seasonal winter rainfall system. So there's a lot more investment in nitrogen early in the season when rainfall is plenty, kind of cool. And as the season progresses, the plants are investing in LMA and more in structure as the summer goes on. But it's a bit more complicated than that. So that's just the general trends. We can actually start to ask questions about how landscape context might influence the allocation of traits as the season goes on. So this is from my recently finished PhD student, Natalie Quayle, and she asked the question in the shift area, whether the legacy of grazing influenced the phenology of traits. So here again is a principal components diagram, and it's showing the different traits that she had going out there. Everything is coded by whether something is ungrazed, moderately grazed, or heavily grazed. Moderately grazed basically means that something has been grazed in recent decades, but maybe not in the few years leading up to when this study occurred. Heavily grazed is continuously grazed. And the first thing that you'll see is that they fill out different trait spaces, but these annual grasslands have a seasonal shift in resource allocation, regardless of whether they've been grazed or not. But the history of grazing affects what those shifts are. And so heavy grazing reflects a shift towards lower nutrients and higher structural investment. And so lower nutrients, higher structural investment, and this kind of makes sense. It's sort of, there's a little bit of an ecosystem memory effect, but also there's probably a species composition effect that's here. So just so that you know, we're going from early season to late season with the bright versus the dark. And then here we're seeing this investment in the heavily grazed in cellulose and LMA, which are going to be related to greater protection and essentially lower palatability to grazers. Okay. What am I doing on time? Okay. All right. So I've gotten through the first two vignettes, which is looking at phenology and the insights we can get from phenology. And the reason why those insights from phenology matter is that models will generally use lookup tables, and then we'll extrapolate across the seasons. And we end up using that to make inferences about things like carbon uptake and resource allocation. And really what this is showing is that if we can go out and make these types of measurements everywhere and look at them phenologically, that the actual trends that we see may vary. The last slide, one of the key points to the last slide is that we can't assume that everything that is the same thing behaves the same. So we can't assume that all oaks or maples or even all red maples may behave the same. We can't assume that we've got the exact same processes going on in these annual grasslands in Southern California, that they differ across these different systems. And knowing something like legacy of grazing, which is shown right here, will help you have better information and be able to better predict how management and then perhaps management interacting with climate change will affect carbon uptake. Okay, so another thing that affects traits is what is actually happening in the system. And I know there's probably a number of folks in here who's actually very interested in plant chemical response to whatever is affecting. The chemical response can be to drought, it can be to herbivory, so insect outbreaks and so forth. It can be related to any number of different processes. And what sort of insights do those traits provide us as to what's going on in the system? I'm going to use two examples here. One is the mega drought in California from 2011 to 2017. It actually kind of continued beyond that, but there was big rainfall after 2017. Then it was dry for several years again, and then there was big rainfall a couple of years ago. But it's a persistent long-term drought. But we have imagery from every year there. And then there's this aspen common garden at the University of Wisconsin, where we've been imaging annually we're now in our fourth year of imaging that. So the California mega drought, this is from the drought monitor shown right here. This is essentially on the y-axis here, it's the percent of California that was in each one of these different categories. So if you look here in starting around in 2013, 2012 to 2014, all of California was in a mega drought. This is where the drought ended, but actually if we extended this on, in this case, I haven't updated the slide, but you can kind of see the drought actually picked back up again here and then it actually got wet again in this year. So a big drought occurred. This is just like, this is the percent of California that was an exceptional drought. This is just remarkable what that drought looked like. And there have been a lot of papers on this, like there was a huge amount of mortality associated with the drought. Actually, the next slide, bark beetles, temperature and precipitation extremes, drought legacies, tree height, so tree hydraulics, leading to greater, taller trees not being able to get water up there, deep soil drying, so trees with deeper roots not being able to access water. There's been a lot of papers that have talked about this mortality and what's behind it. And so we asked the question from this data whether traits are. So we're not actually saying all those other things aren't true. We're just saying, do the traits of the trees that are there, the foliar traits of the trees that are there, give us some insights into the processes that are associated with mortality. And I do want to actually just point out again that a lot of the mortality was caused by bark beetle, but not all of it. Okay, so the site, this is from NASA's Average Classic Airplane, 15-meter pixels. It's been imaged nearly yearly and in some cases multiple times per year. All of these different boxes in California have been imaged. The reason why California gets this is because the plane and the instruments are based at Jet Propulsion Laboratory in the LA area. There are all these different flight boxes. We're going to concentrate on this one right here, which is the Yosemite Southern Sierras box. And that, just for context, that's where the shift box that I talked about, the phenology and the grazing, that's where that was. Okay, so we're going to go in and look at this. We map traits again. And we map them through time. And the general assumption will be that all things being equal, traits don't actually change all that much. They should be considered like relatively constant through time. All of our maps are from the same time. They're from like May-ish, so to the end of the spring wet season in each year. So this is the trait maps. Let's go back here. Actually isn't showing what timing with respect to the drought. So here are the ternary diagrams, much like I showed before, for the trait maps across each one of these years. LMA, so higher LMA means basically thicker leaves. Think conifers have high density of the leaf material. Non-structural carbohydrates, which is this reserve that's used to build other things and is also used as a reserve of energy. And then nitrogen, which is going to be associated with photosynthesis. So if we look across this, 2013 is right at the beginning of the drought. We would use that as our indicator of sort of pre-drought conditions, or just right at the beginning of the drought. And then the drought ended by 2018. So now what you're seeing on this figure here are going to be principal components diagrams. Again, they're all applied to the same full data set. And we're essentially going to look at how the trait distribution changes. You've already looked at all of these maps and already seen that it's different through time. And that's the interesting story. So just starting out with 2013, this is sort of the baseline pre-drought conditions. You see this trade-off like nitrogen LMA. Those of you who know what the leaf economic spectrum is, that's what that is. And then you have these other dimensions that are related to, say, structure, calcium, or non-structural carbohydrates. And the drought started really picking up in 2014 and then really peaked in 2015 and 2016. And so the key here now is that we've moved from being in the middle of this bullseye here oh, I'm sorry, we've moved from our default situation where the middle of the bullseye would be here towards a situation now where there's a lot more investment in the foliage, in phenolics and non-structural carbohydrates. Phenolics are these defensive compounds, non-structural carbohydrates is this reserve pool of energy. I think this makes sense because plants were now becoming very concerned about just basic functions, respiration and so forth. Keeping the non-structural carbohydrates handy in the foliage is a potential strategy for dealing with that. Similar sort of thing. Just note here that this bullseye has now actually moved sort of away from phenolics. So more investment over here in thick leaves, again, good in drought conditions, and non-structural carbohydrates. These are all of the fixes across this whole region. So this isn't separated out into vegetation types, and a little bit of a move away from phenolics. We get a wet winter and things start to normalize a little bit, but not back to where it was at the very beginning. So you actually start to go back and start to see more investment in phenolics, and ultimately you'll start to see levels of nitrogen start to increase. By the way, in 2018, it got dry again. So there is a little bit of wavering that's going around there. And we actually have data from recent couple of years, but we haven't analyzed it yet. So we can go and sort of see that traits are not constant in time and that there's a reallocation of resources that occurs. And so any assumption that traits are constant in time has to be modified by this sort of information. So if we look at this graphically here, this is just the relative amounts of, say, chlorophyll, nitrogen, non-structural carbohydrates. You'll see the chlorophyll actually isn't all that interesting or diagnostic of what's going on in the system. Nitrogen declines, much less access to nitrogen. The trees are slowing down their metabolism, not concerning so much with growth. Increase in non-structural carbohydrates used as this reserve pool. And then phenolics, generally the defensive compounds, decline over the course of the drought, which I would use as an argument for why you saw some of the mortality, is that with the decline of defensive traits, you open yourself up to insects. And even though phenolics aren't important for defending against bark beetles, they're going to be correlated with the allocation of resources to defenses. So I'm going to bring neon back into it. If you look across entire North America, you'll see that non-structural carbohydrates and phenolics are strongly positively correlated. So if times are good and you're able to produce a lot of phenolics, you also tend to have a lot of non-structural carbohydrates in your foliage. But what we see during the drought, non-structural carbohydrates increase while phenolics decline. So this is an important part of the story, is that you're changing your resource allocation strategy. On kind of a global scale, this is the relationship between non-structural carbohydrates and phenolics. But when you have a perturbation like drought, that there's this change in resource allocation. These are density plots across that study area that are associated with the response to the drought. So there are numerous drivers of mortality during drought. So I've just shown you sort of observational results there. Now we're actually going to tie this back to mortality data. So Natalie Quayle, who did the study that looked at the grazing study, also assembled a whole bunch of data about mortality from actually 3 different sources. They all provided their data to her and intersected that with our trait data to ask the question, do certain trait, and the people who had the data had data about when trees died. So she took the pre-mortality trait data and asked the question, is there a relationship between pre-mortality traits and which trees died? And the answer is that there is. So in order to make sense of it, we had to split it out by groups. So mixed conifer, ponderosa pine, and then some of the more broadleaf type species. And so, and the relationship is different across the different taxonomic group. So those figures that I was showing you before, they're actually masking a lot of different relationships that occur across these different types. So there's a higher, in conifers, there's a higher investment in photosynthetic capacity for the conifers that survive versus the conifers that die. This is actually the inverse of the leaf economic spectrum. You would expect there to be more investment in thicker leaves to basically hold on to your water and so forth. And whereas what we seem to have found is that higher investment in nitrogen or lower LMA, higher nitrogen seems to be associated with lower amounts of mortality. In these live oaks and chaparral, the relationship is much more related to leaf hardiness, that there's a greater, so cellulose, which is very similar to LMA, and investment in sugars tend to, greater amounts of cellulose and also higher amounts of LMA, lower mortality. If there was an investment or an accumulation of sugars, which are this reserve margin, trees trying to determine what they're going to do and build with it, actually led to higher mortality. So different relationships across these different taxonomic groups. And that's those are some interesting insights. I think that in even more drought stress systems like pinyon pine, you would probably find different relationships than what we found. But I think a lot of what we found there supports what people might expect in responses. Okay. So now we want to look at traits and responses to insects and insect defoliation. This study is being conducted. This is now at a common garden, WISASP, where there are, which is quaking aspen, 1,824 individuals, actually quite a bit fewer than that now, across 516 genotypes. And we have been imaging it weekly to bi-weekly since 2021. And the reason why we did this is that this was set up by Rick Lindroth. John worked there on his dissertation, right? Did you a little bit? No, he didn't. Okay. Rick Lindroth is a chemical ecologist, very interested in defensive strategies in aspen, and much of it would be related to herbivory. And in 2021, we noticed a lot of egg masses on the trees there of the spongy moth, which led us to actually want to do an experiment to see whether there was actually some genetic drivers associated with defense against herbivory. And we can't do the chemistry on all 1,824 species, but if we can map the foliar traits that are related to defense, as well as things like nitrogen and non-structural carbohydrates, we can actually get that information. It's going to be shown using the imaging spectroscopy data to estimate the different traits. I'm not going to show you the trait models, but we first figured out that we could do this as part of his postdoc many, many years ago. So this is actually just showing from 2021 over the course of the growing season, and you can actually see the defoliation occurring and then the refoliation that occurs afterward. And this is actually just a drone shot of the stand actually at the peak of the defoliation. So you can see that there's some spatial structuring to the way the defoliation occurred, but ultimately everything got defoliated across this system. And this is actually showing you from LiDAR. We have a VTOL LiDAR that we flew that we could also use to sort of get LMA. And basically in 2021, you have this sort of pre-herbivory, and you have the peaks of the herbivory in June, and followed by the refoliation later in the summer. So the question is, how does the chemistry change following defoliation? So there's this sort of constitutive pre-herbivory amounts of defensive compounds. Then do the trees remember that and do they remember that in subsequent years? So we collected hyperspectral imagery. And across all of these different years, what you're seeing are the dates of the images across the season, the dates of the chemical sampling across the season. This was a whole bunch of us came together to figure out how to do this, because this was just one of those opportunistic types of analyses. We built a model. We can estimate these foliar traits. What are the foliar traits that we're looking at? We're looking at pools of phenolic glycosides and condensed tannins. Really generalizing on this, phenolic glycosides are generally tended to be interpreted as defenses against herbivores. Condensed tannins are generally interpreted to be defenses against pathogens. So there's an investment strategy that's going on between building phenolic glycosides and building condensed tannins. Are you afraid of herbivory, or are you afraid of pathogens? So it is going to be really hard for me to figure out how to distill this down, but this is actually the trends of across all. So remember that some of this is genetically determined, so not all trees behave the same. So this is the average across all 1,800 trees. What you're seeing is sort of the average of these traits and then the standard deviation around it. And you're seeing some really messy diagrams showing condensed tannins, phenolic glycosides, nitrogen, and leaf sugars. And so I'll just point out, like, really quickly, one of the stories is that condensed tannins are generally higher than phenolic glycosides, but after the defoliation, there was a higher relative investment in condensed tannins rather than phenolic glycosides. And across all of the years, sugars increase towards the end of the growing season. But this is really a hard way to look at the data, so I'm going to try and show you a couple of different ways. So we're going to go back to our old favorite principal components analysis, and we're going to look at each year and look at the strategies over seasons across years. The years fell out of that. And so this is just looking at these 4 different traits, and I'm going to try and provide some interpretation that we can use to see how the trees have responded. I think the key response is that trees start allocating their resources differently following the defoliation, and they remember it in subsequent years. So looking at the first year, so what we've got, everything here is scaled by, oh, it's hidden. This is scaled by growing degree days. So the darker blue is earlier in the season, and the lighter yellow is later in the season. So you can interpret these graphs of all of these trees across the growing season. So this is early growing season. So one of the interesting things is that Rick and Amy Trowbridge, her collaborators on this, have identified that there are a couple of different chemotypes within this. And so you're seeing at the beginning of the season in this year where the defoliation occurred that there are a couple of different chemotypes, and they really come out on the diagram there. And then you'll see that as we move later in the season, so on refoliation, so this is the middle of the season, there's a movement towards the investment in phenolic glycosides. Trees remembered that they were just defoliated. They're saying, oh, I'm going to shift to phenolic glycosides. Late in the season, resources tend to be sugar reserves in the leaves. I don't have any interpretation for that right now, but that actually happens in all years. You'll see in all of these diagrams. So the trees are defoliated, they refoliate. The next year they refoliate. We ask the question, do they remember that? I would say that it looks like they did, because one thing that's happened in the second year is that the chemotypes have all converged. They all appear to be behaving similarly in terms of their relative allocation to condensed tannins versus phenolic glycosides. I want to actually show this trade-off. It's actually present in all of the different years, but there is definitely a trade-off between photosynthesis and growth and investment in condensed tannins as defenses against pathogens. And then in the third year, the chemotypes are starting to reemerge at the beginning of the season. There's a lot higher investment now in condensed tannins relative to phenolic glycosides, which you can actually just see right here with this blue line the condensed tannins line being higher than the red line, the phenolic glycoside line, whereas they were a lot closer over there in the first year. Why is that? Well, when the trees started investing a lot more resources in phenolic glycosides following the defoliation in 2021, they decreased their relative investment in condensed tannins and opened the trees up to exposure to pathogens. And we just saw that, and we see that in the trends that we see in the data, that condensed tannins declined just for a little bit. They picked back up in 2023 and probably into 2024, but that was enough time to be able to let some pathogens get in. I'm not exactly sure what the specific pathogen is at this point. We've got a student who's going to be, there's multiple pathogens that are present, is part of the story that's going on there. And so the phytochemical shifts compromise the resistance to pathogens. And that's why I said it was 1,824 trees to start out. It's probably 1,700 trees now because we've had a lot of mortality, and they were probably in the chemotype that was invested more in condensed tannins and switched to phenolic glycosides. So I am almost out of time. And so I'm going to skip over the memory effect, but we did just because it's going to be very hard to explain. So we're actually trying to test what the memory effect is using each tree as its own observation. And it's actually just, I can maybe just show this. It's like phenolic glycosides have a long memory of their initial starting condition, of what their constitutive state was before they had a defoliation, and then have a short memory. That means they're really highly variable across the season in post-defoliation. Nitrogen has a very short memory, and that's just because nitrogen changes across the season. That's going to be true no matter what the case is. And so condensed tannins don't have, there's no really difference in the memory of condensed tannins in these systems before or after. So it was actually the relative concentrations of phenolic glycosides versus condensed tannins rather than the absolute. We can also actually look across seasons. Condensed tannins, there's no difference in the sort of memory effect seasonally versus annually. So this is across years and across seasons. No difference in phenolic glycosides. So they kind of behave the same whether you're looking at it across years, but nitrogen has a short memory seasonally, sugars have a long memory seasonally. Condensed tannins have a shorter memory, and phenolic glycosides have a longer memory. That's just this being kind of closer to zero long memory, lower value of alpha is a longer memory. So, okay, but I really wanted to just move on to just talk really quickly. I think I can sneak it in here, John, about ecosystem function. My main interest with all of this has actually been to link to carbon uptake. The ways that we measure carbon uptake at the leaf level might be with something like an LS6800, but I'm going to talk about flux towers right now. So you have a flux tower, and there's a footprint dependent on the wind direction. And so actually the wind direction would be like this. Here's the flux tower making all of these eddy measurements from which we estimate GPP. The characteristics of the vegetation within this footprint will tell us something about what the uptake is. And there's an airplane flying over and making our trait measurements. So what the flux tower is measuring is this complex model, but there are a whole bunch of components of it in the leaves and the carbon content, nitrogen content, and trade-offs that we can use to within each one of these flux towers. So going back to neon, we've mapped traits across all of the neon domains. All of the neon domains have flux towers, and we can look at the trait variation within these flux tower footprints, which are shown here. We can calculate these footprints on a seasonal basis or on a daily basis, and we can look at how, as the flux tower footprint changes, we can estimate the trait variation. So the basic process is we map traits, We can look at GPP from the flux tower across all of these dates. We can look at nitrogen within this ever-changing footprint. The footprint is changing because wind directions change. We model the footprint, and we can start to say, ask the question, do the traits actually influence what's going on? For the most part, people use leaf area index and then climate measures like temperature and vapor pressure deficit to estimate carbon uptake. We're just saying that traits might provide additional information. And this is actually just looking at daily morning GPP and footprint weighted nitrogen. So I'm not even controlling for LAI or climate variables. And actually, we can just see from the outset that there are significant relationships in some domains just between the traits. So what I'm saying is that actually, if you know LAI and climate variables, you can do a pretty good job of estimating GPP. In some places, such as Harvard Forest, Steigerwald, which is in Wisconsin, and University of Kansas Field Station, there are actually some pretty significant relationships, but the relationships are different. And it's not, I showed nitrogen because that's most related to carbon uptake, but then you can look at things like phenolics and non-structural carbohydrates, and it becomes a little bit more complicated in terms of thinking about resource allocation trade-offs. And then you can go to other sites, such as Harvard Forest from the 2019 data, and you can find no relationships at all. And in fact, LAI, temperature, and vapor pressure deficit may do a better job of explaining it. So if we look at the residual variance after we control for LAI, temperature, and vapor pressure deficit, and start to look at traits again, then their partial correlations become significant, and they're all in the direction that we would expect. So nitrogen and phosphorus have positive relationships with GPP after controlling for LAI, and climate, LMA, well, non-structural carbohydrates, again, that will change directions on different sites. Phenolics generally doesn't have any relationship. Phenolics actually doesn't have much to do with photosynthesis in this case. And so we can look across all of these deciduous broadleaf, evergreen needleaf forests, mixed forests, and grasslands, and we can see that once we control for these differences in climate that would occur across all these different biomes, we have these strong predictive relationships between traits and GPP. So this tells us that we're not just, we can actually start to take what we learn at flux towers and we can expand it out more broadly and probably develop better models of GPP, gross primary productivity, if we have this spatially varying trait information. And this is just actually showing you how a control model is improved by nitrogen. So it's a modest increase in the predictability of the model, but it's still something, and it might help close some of our gaps in knowledge. I just wanted, I took the slide out. The question could be asked, why not use NDVI or other vegetation indices? Those actually didn't improve the models at all. It was actually the traits derived from the spectral information that improved the models. Okay, so just my final thoughts. So this is from a very famous paper by Sandra Diaz, the global pattern, the global spectrum of plant form and function, that was in Nature from 2016, showing data from TRI, the database, showing the distribution of traits on a global basis. This is from NEON, from our data, and it's showing the same patterns. The traits are different, so we don't have tree height. We could get tree height. We don't have seed mass or wood density, but we have different variables, and some of these actually may correlate with those other variables. So we can actually characterize these global patterns using remote sensing data that people get from field measurements. And so that led people to ask me, so what is the leaf economic spectrum, how would I say it? The allocation of resources. What is the remote sensing based allocation of resources? So there are things that we can't measure from remote sensing, but what is it that we can measure? So the leaf economic spectrum is this sort of famous thing that came out of a number of papers in the late 1990s and early 2000s that basically describe plants globally as allocating their resources between higher nitrogen and higher LMA or lower SLA, which would be investing in growth versus investing in leaf construction costs. So you do this when you think you're going to lose your leaves, or when you have seasonal drought, or you live in a tough environment. This we can absolutely do from remote sensing. We can also do defense remote sensing. So there's another dimension to this imaging spectroscopy leaf allocation, which I call phenolics here, but also things like lignin and cellulose that make leaves be not palatable also show up on it. The cool thing is that you also have these multifunctional traits, non-structural carbohydrates, which might be used for maintenance or reserve safety margins or whatever, which is yet another dimension that may actually correlate with all of these and play roles in all of these, but is another unique dimension in and of itself that seems to be indicative of responses to stress and changes over the course of the growing season. Okay, so that's it. This is just a reminder that this is remote sensing. This again is David Hockney here. This is the painting and showing you how he did it. I kind of like to use this as a metaphor of like what we're doing with remote sensing is still just making a painting of something that we're trying to interpret it. So actually getting out into the woods here actually gives you the information that you want. But what we're trying to do is just build up this story from these different components that we can pull from this. And I hope I've given some insights into it. And the main reason why I do all of this is I get to go to beautiful and interesting places. So thank you very much. Yeah, sorry.
I was curious if leaf hydration impacts the spectral signature that you might get from trait. So I was thinking about your drought work in California with the increase in leaf MSCs. I'm wondering if it's an actual increase or you might just see it as an increase because other people of water content.
Okay, so I pulled out, I think I kind of let sneak in there in a couple places the water content. So we can get water content concurrent with the images. So we can get leaf water content. The hard part about leaf water content and phenolics, to a lesser extent pigments, is that they can change quite a bit diurnally, and there's this dilution like that. So my answer to you is I think that's a good question, and that begs a little bit further analysis in there. But we generally tend to set the water content part aside just because that might be so dependent on the conditions that occur at the time of imaging. Yeah, not a satisfying answer, I think.
But wouldn't another sensor that could actually, focus on that potentially be complementary?
Yeah, I didn't
The
even.
absorption features for water, might be unstable, but I think at a bigger scale, some of the other instruments.
Yeah, and I actually showed Bina, I think, just a couple of slides from this. If we can integrate thermal sensing, we can actually see a lot that's related to stomatal closure and evapotranspiration and related to water content as well. Yeah. And I just went through and showed you everything that was hyperspectral remote sensing, tended as if no other sensors like LiDAR even existed.
They treat us the same way, so it's okay.
I think there's some people stigma questions, but thank you very much for